Thanks for being in my talk titled Vocal Sequences Emerge Slowly in Chimpanzees Development. One way to understand the origin of human language is looking at the proximate level. For example, vocal development. So we know that in humans, for example, children develop a unique and complex combinatorial capacity through vocal learning and neural maturation. However, in primates, our close living relatives, most studies today have looked at acoustic changes or vocal usage through ontogeny, but little is known about the development of vocal sequences. A recent study in chimpanzees showed that adults present a potential complex vocal system with an extensive use of sequences across the whole vocal repertoire. This makes chimpanzees a good model to study the development of vocal complexity. So, the goal of our study is to analyze the development of vocal structural complexity in wild chimpanzees and investigate which processes are involved. Here, with vocal structural complexity, we refer to the combinatorial potential and to the production of utterances that might be harder to produce, requiring more coordination of articulations and muscles. Based on previous studies, our main hypothesis states that social complexity drives vocal complexity. This means that species or populations living in a more complex social group require a more complex signaling system. So most of the studies to date have looked at this in a comparative way. However, here we will use the expansion of the social world and the integration of the chimpanzee in the community through development as a proxy for increases in social complexity. So we have here the timeline of the main developmental stages of chimpanzees, which shows two key ages where social contacts unfold. The first one is at weaning age, that is around 4 years of age, where chimpanzees are no longer being carried by the mothers, and this leads to a more independent engagement with adults of the community and social communication of food and danger. The second important period is the sub-adulthood, starting around 9 years of age, where the association with the mother drops and they become socially active members in this complex fusion society. So according to this, we predicted vocal complexity increase with the steepest increase around weaning age and reaching the other levels of complexity at the beginning of the sub-adulthood. We have as well three alternative hypotheses: limited maturation, neuromuscular maturation, and sexual selection, and each of them has a distinct ontogenetic trajectory. So I went to the field to record all vocalizations from the habituated chimpanzees living in the Taï National Park in Ivory Coast. I got around 1,500 hours of vocal data, from which I got more than 10,000 vocal utterances from 98 chimpanzees range from 0 to 55 years old. I coded the data visually inspecting the spectrograms. Here we have an example of a chimpanzee vocal sequence, which is formed by 7 distinct vocal units, and some of the vocal units can be produced with voice inhalations in between. So for example, this panted bark, where we can see a line in the spectrogram that is marked with the arrow between the two barks, and this is why we call them a panted bark instead of just a bark. So once we coded all the data, we developed generalized nonlinear models which allow us to detect the inflection point and the asymptote of our response variables that are related to vocal complexity, which are the maximum utterance lengths. So in the example from the spectrogram before, it would be 7, the probability of an utterance to be panted. So if the utterance presents at least one panted unit, the probability to produce 2 adjacent pants, so for instance a panted grunt followed by a panted bark, which might be anatomically harder to produce, and the utterance diversity, also called repertoire size, that is the proportion of unique utterances given a total set of utterances. We include as well as a test predictors age and sex, and as a control variable the dominance rank. Moving to the results, we have here the maximum utterance length in the y-axis as a function of age in a log scale. Each data point represented data of one individual and study period. In blue we have males and in red we have females, and the line and polygons are the fitted model and confidence intervals. So we found that the maximum uterine length increased through development and with similar trajectories for males and females. The steepest increase is around 4.5 years of age, and the adult levels of complexity are reached around 8 years of life. When looking at all our variables, we found a similar trajectory with increase through ontogeny of vocal complexity and no sex differences. 3 of our variables present the steepest increase around weaning age and adult levels of complexity in sub-adulthood between 8 and 10 years of life. These results support our predictions for the social complexity hypothesis. However, when we look to the probability of producing 2 adjacent bands together, we see a similar pattern, but this seems to be shifted later on ontogeny. So if we zoom in the probability to produce 2 adjacent bands, we see that the emergence is at 4 years of life, with the steepest increase around 7-8 years, and reaching adult levels of complexity around 12 years of life. Although these results do not fulfill our predictions for the neuromuscular hypothesis, the production of 2 adjacent bands might be a further layer of vocal complexity that might require higher neural and muscular maturation. So, taking all this together, our results suggest that chimpanzees present multifaceted processes regarding to vocal development, with neuromuscular maturation and social complexity driving vocal complexity. As a summary, chimpanzees need 10 years to develop the vocal structural complexity present in the adult vocal repertoire, and this is way beyond the age of immersion of single units, which is at 4 years of life. And these results contrast the limited and inflexible vocal repertoires present in many primates. Besides, like in humans, the increase in vocal complexity through development is likely driven by a multifaceted process, neuromuscular maturation, and increase in social complexity. Regarding the social complexity hypothesis, exposure to greater social complexity through ontogeny might develop the ability to combine single units into complex sequences to expand message potential. Whether there is learning in this acquisition remains to be studied. Our results support the idea that social complexity might promote the evolution of complex vocal repertoire as an ultimate explanation for human language evolution. Thank you for listening, and thanks to the collaborators that made this project possible.